Today we're going to extract DNA from strawberries. This experiment will show us how we can isolate the genetic material found in living cells. Materials needed for DNA extraction: fresh strawberries or another soft fruit, a plastic Ziploc bag, dish soap, table salt, water, a beaker or measuring cup, a funnel, a coffee filter or fine strainer, test tubes or clear glass, ethanol, ice-cold rubbing alcohol, and test tube rack. First, place the strawberries inside the bag and seal it. Then, mash the strawberries to break down their cell walls. Since strawberry skins are tough, this requires a bit of persistence. To prepare the extraction solution, add the salt and dish soap to the water, then stir gently until the salt dissolves. Next, we add an extraction solution made of water, soap, and salt. The soap dissolves cell membranes, releasing the DNA into the solution. Now we filter the mixture. This separates the large cellular debris from the DNA-rich liquid we need. Finally, we add ice-cold alcohol. Since DNA is insoluble in alcohol, it precipitates out as these visible, white, thread-like strands. And there it is, real strawberry DNA. A simple yet powerful way to see the building blocks of life. Let's test your knowledge with some review questions. Question 1. Why are the strawberries mashed? A. To cool them. B. To break apart the tissue and cells. C. To remove the DNA. Or D. To change their color. The correct answer is B. To break apart the tissue and cells. Question 2. What is the role of dish soap? A. It dissolves cell and nuclear membranes. B. It colors the DNA. C. It cools the mixture. Or D. It filters the extract. The answer is A. It dissolves the cell and nuclear membranes. Question 3. Why is salt added to the extraction solution? A. To make the mixture sweeter. B. To help the DNA strands clump together. C. To dissolve the coffee filter. Or D. To warm the solution. The correct answer is B. To help the DNA strands clump together. Question 4. Why should the rubbing alcohol be ice cold? A. Cold alcohol helps DNA precipitate. B. Cold alcohol dissolves DNA. C. Cold alcohol changes DNA to red. Or D. Cold alcohol breaks the test tube. The correct answer is A. Cold alcohol helps the DNA precipitate. Question 5. Where does the DNA accumulate? A. At the bottom of the strawberry extract. B. Inside the coffee filter. C. At the boundary between the strawberry extract and the alcohol. Or D. Inside the salt. The answer is C. At the boundary between the strawberry extract and the alcohol. Question 6. What does extracted DNA look like? A. Blue crystals. B. White stringy material. C. Red powder. Or D. Clear bubbles. The correct answer is B. White stringy material. Question 7. Why should the two liquid layers not be shaken? A. Shaking may mix the layers and make the DNA difficult to observe. B. Shaking freezes the alcohol. C. Shaking changes the strawberry color. Or D. Shaking removes the salt. The answer is A. Shaking may mix the layers and make the DNA difficult to observe.